So hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj sir, your biology master teacher. Students, welcome back to your own channel students. Now students, you tell me in the chat right now, every single question, every single mark in your examination is very important. All of you will agree with me. A single mark can change your rank in thousands, let alone a question, one question which has 4 marks that can completely change your fate. Tell me yes or no, students, tell me yes or no. A single question which holds 4 marks can make you into a doctor or make you into a dropper. A single 4 marks or a single question, students, a single question. So students, you tell me in the chat right now, is it possible to leave those 4 marks for granted? I'm like, what is it? Just one question, no? One question. What is so, difficulty? One question I will leave. One question I will leave. What is the big deal? You cannot have that attitude at all. You cannot have that attitude at all, students, because a single mark can completely change your future, right? Now, students, if you have noticed in your previous year NEET questions, it could be last year, a year before that, or year before that, every single year, one question has always come from the scientist or the dates. So students, you tell me, that is how important this session is, because this session right now will make sure that you do not lose that at least 4 marks. Very important, 4 marks to go. Okay, students, all of you in the chat, quickly let me know in the chat right now if I'm audible and visible to all of you. Yes, students, quickly let me know chat right now if I'm audible and visible to all of you, students. Hello Dilip, welcome. Yes, welcome students. Quickly let me know chat right now if I'm audible and visible to all of you, students. Yes, more than one, sir. Exactly, that is how important a single question or a single mark is, students, because one single question, if you get wrong, right, your rank will change in thousands. It will change in thousands. So you cannot afford that. You cannot afford it, students. So this is a session which will teach you the importance of scientists and importance of dates. Now again, students, you might ask me, sir, why do I even need to learn the name of a scientist? Students, understand, if these scientists did not work in their time, the advancement in science would not be possible. These scientists laid the foundation work for all the luxury which you are enjoying right now. So to honor these scientists, to know the year which they worked is very, very important, very important, students. So with that being said, students, here is the first list of scientists which you need to know. The first scientist is called, students, all of you, every single scientist, I want everyone to do a namaskara in the chat, because I want us to pay respect to our scientists. Yes or no, students? Yes or no? So here I want all of you to put namaskara to all the scientists here for all their hard work throughout the years. Now students, the first scientist right here on the board is called as Ernest Mayr, who is also called as the Darwin of 20th century. Darwin of 20th century, right here, students. Now, throughout his nearly 80 years of career, his research spanned from ornithology, taxonomy, zoogeography, evolution, systematics, and history and philosophy of biology. So he single-handedly made the origin of species diversity and central question on evolution biology that is today. So he was a scientist along with Darwin, laid the foundation of our understanding of our entire evolution. If this scientist did not exist, the entire evolutionary we would not be able to understand so well. So I want all of you to put namaskara quickly in the chat, all of you. Ernest Mayr, OP in the chat. Now, second is your Carl Linnaeus, obviously. The importance of Carl Linnaeus in binomial nomenclature, taxonomical hierarchy is unmatched. It is completely unmatched, students. Carl Linnaeus, the scientist who gave us binomial nomenclature, the scientist who gave us the entire taxonomical hierarchy, kingdom, class, order, family, genus, species, the entire hierarchy was given by Carl Linnaeus, students. Okay, so he gave us the binomial nomenclature. Generic name is the first, species epithet or specific epithet is the second here, students. Second, it is called as binomial nomenclature, students. Can I ask all of you a simple question? The question is, students, in binomial nomenclature, what is the origin of the letters here or the words here? If the origin is something different, it is converted to one particular language. Even if the origin is different, the words are converted to a particular language, students. What is that language? Anyone who can tell me, students? Anyone who can tell me which is the language it's converted to? Students, all of you? Students, exactly, students, they are so important. Students, this is the importance of speech. You will learn throughout the history, giving speeches. Okay. Now then we have George Bentham and Joseph Jardine Hooker. right? The natural classification system, students, these two scientists gave the natural system of classification. Carl Linnaeus, artificial again. Carl Linnaeus again, artificial. Carl Linnaeus again, artificial. Remember that much. Okay. So, what do you mean by natural system of classification? In natural system of classification, you look at the inside beauty. You look at how are organisms from the inside, not the muti on the top. Okay. So, look at the anatomy, embryology, cytotoxin, cytology also. So, you look at the internal beauty here. Okay, so natural system of classification, internal features like your ultrastructure, anatomy, embryology, and phytochemistry is used in your natural system of classification. Now, students, next scientist is called as Carl Linnaeus again. Students, again, he is coming again and again. Okay, artificial system of classification, and also gave the book Systema Naturae. Book was written by. Then Aristotle, two kingdom classification. R.H. Whittaker gave the five kingdom classification. Students, I want every single student to tell me which are the five kingdoms in the chat right now. Which are the five kingdoms? Kingdom Monera, Protista, Fungi, Plantae, Animalia. Five kingdoms in the chat, all of you. Five kingdoms in the chat, all of you. Quickly, namaskara to R.H. Whittaker again. Now, students, again, 3 kingdom, 4 kingdom, 6 kingdom, not required. They will not ask you from there. Just remember the entire, you have to remember the 5 kingdom mainly. 5 kingdom mainly, students. So, students, also tell me in the chat right now, out of these 5 kingdoms, in which kingdom do you find unicellular eukaryotes? Under this 5 kingdom classification, which organisms are unicellular eukaryotes? Unicellular eukaryotes also. Now, next one is Dmitry Ivanovsky, students. In 1892, students recognized certain microbes as causal organisms of TMV, tobacco mosaic virus. That was your Dmitry Ivanovsky. Now, students, again, if you want this PDF, the PDF will be available on Telegram channel. It will be available on the Vedantu NEET English Telegram channel. It will be available on Basavaraj Biology and also Biology by Gopika. Students, Biology by Gopika, right? Gopika, ma'am changed this thing, right? Students, ma'am changed to official something. So official Gopika ma'ams. Okay, don't join anywhere else. Okay, so official Gopika ma'am also will be available. Okay, the entire PDF. Now, then we have M.W. Benjamin. In 1898, he told us that, students, he told us that, for example, one plant is there. This is one plant here, right? This is one plant here, and let's say this plant is infected here. This plant is infected, right? Infected plant. Now here we have one more plant here. Here we have one more plant. This plant is healthy plant. This plant is healthy plant right here. Now what if I take the extract? I take the extract of this plant and inject into this plant. What will happen? This plant will also get infected. This plant also will get, students, what? Infected completely. That is called as what? That is called as contagious fluid. That is, students, demonstrated that the extract from the infected plant of a tobacco could cause infection in healthy plant, and name the new pathogen as virus. Name the new pathogen as a virus here, students. Virus as a new pathogen here. Okay, and call the fluid as Contagium vivum fluidium. Your Harry Potter spell here. Okay, Harry Potter spell, students. Contagium vivum fluidium. Infectious living fluid. So infectious living fluid was given by none other than M.W. Benjamin. Very, very important, students. Very, very important. Contagium vivum fluidium. Okay, Harry Potter spell right here, students. Now again, students, here T.W. Ivanovsky also told us that viruses are much more smaller than bacteria. He also told us that viruses are much kuti. They are very, very small. They are compared to bacteria. Next, students, next. Then we have W.M. Stanley in 1935. Students, years, again I'm telling you, years are not so much asked compared to the scientists in the contribution. Scientists in the contribution is actually much more important than the years here. So I will strongly suggest that the first priority of understanding has to be the work here, then the year. Okay, students. Now, he showed that viruses could be crystalline, and crystals consist of largely of proteins. They are inert outside the specific host here. Viruses are obligate parasites. That is, they cannot live without a host. They cannot live without a host here. Okay. Now, T.W. Diner, who told us that about viroids. He told us about viroids, students. Students, can anyone tell me in the chat right now? Anyone in the chat who can tell me? The viroids, do they have only RNA or only protein? In the chat quickly. Do viroids have only RNA or only proteins? Which is the case in the chat? In the case of viroids, do they have only RNA or only protein? in the chat. Tell me, students, in the chat, all of you. Now, next, George Bentham and Joseph Hooker, natural system of classification again. See, natural system of classification. Now, then there is Catherine Sau. This scientist's name is given just before the morphology chapter, structural organization. Before the structural organization, the name is given. Okay, the scientist, she reported her in her early publication that curly top virus spread through the plant via the food conducted or phloem tissue. So basically, the scientists discover entire work based on the entire transportation or the entire flow of xylem and phloem. Okay, the fluid conducted or phloem tissue took a dynamic development approach designed to enhance one's understanding on plant structure and enormous impact worldwide. So the scientist's main work was on plant anatomy, students, plant anatomy. Now then we have G.N. Ramachandran. All of you know Ramachandran plot, right? Ramachandran plot allows us to understand the structure of a protein, and this is a scientist who is also called as Indian biophysics, father of biophysics in India. Okay, his discovery on triple helix structure of collagen was very important, because back then everyone used to think alpha helix, beta plated, but because of Ramachandran, we got triple helix model, and also the protein structure, Ramachandran plot. With the help of Ramachandran plot, we can study the structure of the proteins. Then Anthony von Leeuwenhoek, students, Anthony von Leeuwenhoek first saw the described living cell, students. Can anyone tell me in the chat right now which was the scientist who discovered dead cell? Anyone in the chat? Students, why will they laugh at you, students? Why will they even laugh at you, students? Scientists are proud that the students right now, the current new generation of students that are pursuing science, their work is still being used. So from the heaven, they'll be smiling at you, all of you. Okay, friends, tell me in the chat. Which scientist discovered the dead cell first time? Which scientist discovered the dead cell, students? Dead cell. All of you, students. All of you in the chat. Robert Hooke. All of you. Robert Hooke, students. Robert Hooke. Okay. Then we have brown color. Robert Brown. Okay. Discovered nucleus in the orchid, right? Orchid root. Mathis Scheiden and Theodore Swan. Both of these scientists worked on what? Worked on cell theory. Now, students, here is a small change. Can all of you see a small change here, students? Can all of you see a small change here? German botanist, German zoologist. Students, in the old NCERT, what was it, students? In old NCERT, what was the issue? In old NCERT, here it was British zoologist. Tell me yes or no, students. In old NCERT, it was British zoologist. But right now, according to new NCERT, it has been changed. According to new NCERT, it has changed, and all of you should be aware of it. So in new NCERT, it's supposed to say German zoologist. German zoologist. Please remember that, students. Please remember that. Now, first of all, the botanist. Obviously, he'll work on plants. German zoologist, students. Theodore Swan told us that around there is something called a cell membrane. In the case of plants, it has a separate layer also called as your cell wall, is present only in the case of plants. And together they gave a cell theory known as every single animal and every single plant, we are made up of cell and product of cell, students. We are made up of cell and product of cells. That was cell theory. Now, students, there was one more scientist who came and told us, all of you do not know anything. I will tell you how cells exist, how the cells come into existence, students. Who is the scientist who told us that cells exist from a pre-existing cell, students? Who was the scientist? Who was the scientist who told us that cells come into existence from a pre-existing cells? That was Rudolf Virchow, students. Rudolf Virchow in 1855 told us that cells exist from pre-existing cells, and he gave a statement, omnicellula e cellula. Again, Harry Potter spell. Again, a Harry Potter spell right now, students. Okay, Harry Potter spell right now. Okay, so omnicellula e cellula. Now, Singer Nicholson gave the fluid mosaic model. All of you should know. Camillo Golgi discovered the Golgi body. George Paldane, ribosomes, discovered the ribosomes. Melvin Kelvin, friends, Melvin Kelvin sounds like a chocolate name. Oh, sounds like a chocolate name. Friends, sounds like a chocolate name right here. Melvin Kelvin. Okay, so Melvin Kelvin, Kelvin cycle. Melvin Kelvin, Kelvin cycle. Kelvin cycle, students. All of you should know Kelvin cycle. Friends, is the Kelvin cycle called as C3 or C4? Kelvin cycle is called as what? Kelvin cycle is called as C3 cycle. C3 cycle here. C3 cycle. Okay, he gave us the Kelvin cycle right now. Joseph Priestley, students, Joseph Priestley, he told us that oxygen is important for, oxygen is very important. Oxygen is very, very important, students. Okay, oxygen is very, very important now. Now, again, students discovered oxygen. Students, imagine, before 1774, what did people even breathe? Students, do you know that meme, right? You know the meme, right? Before 1774, I don't even know what people breathe, because until then oxygen was not discovered. Okay, so in 1774, Joseph Priestley discovered the oxygen. Who knows what they were inhaling before? Okay, someone is saying Calvin Klein. Students, Calvin Klein is different. Why are you bringing Calvin Klein here? Okay, now Jan Ingenhousz, students, showed that the sunlight is essential for plant process. Jan Ingenhousz, students, his name only is an engine. Can you see engine house? It's an engine and a house, like a train engine of the house. Jan Ingenhousz is an elegant experiment with an aquatic plant showed us that the bright sunlight and small bubbles were formed around the green plants. That is, the entire process of photosynthesis happens in the daytime, not in the night time. Simple as that. Okay. Then Julius, students, see the names. Why are the names so fancy and so good? Julius von Such, right? Julius von Such, he provided evidence for the production of glucose when the plant grows, and glucose is usually stored in starch, as starch. So glucose is stored as starch in the case of plants. Then T.W. Engelmann, name only has a man in it. Students, imagine, name only has man in it. Man is there in the name only. T.W. Engelmann. Okay, testosterone level high of the roof. Okay, the first action spectrum of photosynthesis was given by T.W. Engelmann, students. Now, can anyone tell me, students, in T.W. Engelmann experiment, which algae did he take? Anyone? Which algae, students? Which algae did he take? Which algae? Jan Ingenhousz. Exactly. Friends, tell me, which algae did he take? He took as cladophora. And students, did he take aerobic bacteria or did he take anaerobic bacteria, students? What did he take, students? He took aerobic bacteria, and he noticed that all the aerobic bacteria is clustered. I repeat again, students, when he took a light, this was the entire test tube right here, and here we had a prism right here, and the white light was passed through. I'm teaching physics now, okay? And red light and blue light was scattered around. Now, students, can anyone tell me aerobic bacteria was observed here? And all the aerobic bacteria was concentrated in which two regions, students? Can anyone tell me all the aerobic bacteria was concentrated in which two regions? It was concentrated in red and blue region. And it shows that in these two regions, photosynthesis is high or low? In these two regions, students, in these two regions, is the photosynthesis high or low? Photosynthesis is very high. Since in these two regions, photosynthesis is very high, it means there is more O2 in these two regions, and that is why more aerobic bacteria in these two regions. More aerobic bacteria in these two regions, students. In these two regions, more aerobic bacteria. Done, students? Done. Now, then there's Cornelius Van Niel experiment. That is, H plus is the hydrogen donor. So hydrogen is coming from the water, not from anywhere else. Okay, friends. Now, Kranz anatomy, students. Kranz anatomy. Can anyone tell me in the chat? Kranz anatomy and Hatch and Slack pathway. Which cycle are they involved in? C2, C3, C4 in the chat. Kranz anatomy as well as Hatch and Slack pathway both are involved in C4 cycle. Only in the case of your C4 plants we can see something called as Kranz anatomy. Okay, Hans Kranz. Kranz means wreath. What is the wreath? Wreath means like this round structure, wreath. Okay, and it's reflection of the arrangement of cells. The bundle sheath cells may be from several layers around the vascular bundles. So around the vascular bundles, we can see bundle sheath cells around it. Okay, Hatch and Slack pathway is called a C4 cycle. All of you know that. That's what answer. What is the other name of glycolysis? What is the alternative name of glycolysis, students? Alternative name of glycolysis. Glycolysis is also called as EMP pathway. It is also called as, students, what? EMP pathway. Emden, Mayeroff and Parnas. So Emden, E for Emden here, M for Mayeroff and P for Parnas. So Emden, Mayeroff, Parnas, EMP pathway. Okay. Charles Darwin and Francis Darwin, both of them discovery of auxin. Students, Can anyone given here students? Isolation was FW went. Isolation of auxin was FW went. Now question to all of you students. In discovery of auxin, which of the experiment plays a major role? Geotropism, hydrotropism, phototropism. Tell me in the chat, students. Tell me in the chat. Which of the following plays a major role in the discovery of auxin? Phototropism. Students, all of you, if you are new to the video, quickly smash that like button right now. And if you're new to the channel, please subscribe also. Please subscribe. Okay. Now, students. Now, Kurosawa discovery of gibberellic acid. Students, Gibberella fujikuri was the fungus here. Okay. Now, students, can anyone tell me in the case of your gibberellins, what is the main feature of gibberellins? Increase in internodal length. Yes, you're correct. Now, students, Skoog and co-workers discovery of cytokinins. Here. Friends, can anyone tell me what is the main function of cytokinin? Main function of cytokinin is what? Cell division. Main function is cell division. Auxin, root and shoot initiation, apical dominance, parthenocarpy, feminizing effect. Friends, I'm telling you again, the entire information on hormones has to be on tip of your tongue. If I ask you this particular event, this particular feature, you should tell me which is the hormone. For example, hastening of maturity period in conifers, grape stalk length, increase in bolting in rosette plants, which hormone? Now, then we have mineritol, that is identification and crystalline of cytokinins, promoting active substance termed as kinetin. Kinetin was obtained here. Okay. Friends, can anyone tell me again, kinetin is natural or synthetic? Because there is something called as zeatin also. There is something called as zeatin. So, can anyone tell me, is kinetin natural or synthetic? In the chat. Then we have HS cousins. Both are cousins here. Okay. HS cousins confirms the release of volatile substance from the ripened orange that hastens the maturity period in banana also. That is, you take a very ripened orange and kacha mango. Kacha mango means what? Unripe mango. Okay, unripe mango. Now what happens that, or unripe banana you can take. Okay. Now what is happening is this orange, it will release some type of gas. Not the gas you're thinking. The gas here is ethylene gas. It will release the ethylene gas from here. Ethylene gas. And because of this ethylene gas, the entire unripe mango or unripe banana will become ripened directly. Okay then students. Then Panchana Maheshwari students, he worked on embryological aspects and popularized the use of embryological characters in taxonomy. So until Maheshwari did not work, majority of your classification did not even consider embryology until here. Friends, can anyone remind me, in the case of monocot, a single cotyledon is called as what? See, embryology, it came to my mind right now. A single cotyledon in the case of monocot is called as dash. Question for all of you. Now, then we have George Mendel, all of you know students. Namaskar, all of you. Big namaskar quickly students here. Big namaskar right now students. Synthetic, exactly, synthetic students. Now, George Mendel, hybridization experiment, 7 pairs of contrasting traits. Friends, can anyone tell you 7 pairs of contrasting traits means 7 traits, 7 characters, 14 traits. Okay. Hugo de Various, Carl Cons, Eric von Schumacher. I've told you students always, until Mendel's death, his work was not recognized. Mendel died knowing that his entire work was never recognized. Why students? Why? Because back then there was no social media. Right now, if you discover something, you will go to Instagram. Oh my God, I discovered this. You will put a post here. But back then there was no Instagram. There was no internet back then. And let me small fact students. When I was your age, there was internet that time. There was internet. Okay. Now students. Then what a certain Theodore Boveri gave the chromosome theory of inheritance. They told us that behavior of a chromosome and behavior of a gene is parallel to each other. They're same to same. Now, chromosome movement during the meiosis, 1902. Thomas and Morgan, T.H. Morgan, experimental verification of chromosomal theory of inheritance. Now, students, can anyone tell me in the chat, all of you, which was the organism used by T.H. Morgan in his experiments? And T.H. Morgan gave which two terminologies? Which two terminologies were given by T.H. Morgan? Something with link. Okay. Alfred Stewart Went. Alfred Stewart Went is I was going to say son, not son, student. He's a student of T.H. Morgan. He gave gene mapping. He gave us the gene mapping, also called as centimorgan. That is, Alfred Stewart Went gave us the distance between the two genes. Distance between the two genes. Alfred Stewart went. Okay, Alfred Stewart went. Distance between the two genes. Okay, that's what answer. Henking 1891 X body, he gave us that. Punnett, all of you know Punnett square. Langdon Don gave us the Down syndrome. Students, Down syndrome, which chromosome? Which chromosome again? Is it X chromosome, Y chromosome? Down syndrome is X or Y chromosome? No, both are not. Down syndrome is 21st chromosome, not X or Y. In the case of Klinefelter, it is XXY. In the case of Turner's, it is X0. Please remember this much. Okay? Please remember this much, students. Now, F.W. Gauss, I already told you, students, comparative exclusion principle. He told us that, students, when there are two species are competing with each other. Two species are competing with each other. One is superior, it will survive. One is inferior, it will perish. Gauss comparative exclusion principle. Okay. Edward Wilson, students, biodiversity term. Now, this date is very important, students. This date is very, very important here. Very, very important here. Okay. According to students, one more is missing here, that also should remember. Okay. Biodiversity. One. According to International Union of Congress, Natural, that is IUCN, total number of plant and animal species described so far are slightly more than 1.5. Now, students, here all of you should remember the Rio de Janeiro one and the Johannesburg one. Both are very important here, and date is very important. Okay, date is very. This is not given here, so write on that one. That one you have to write down here. That one, students, it's not given. No, students, I thought I wrote it. Oh, it's given here, students. Given here. It's given. Sorry, it's given here. 1992 and 2002. It is given here, students. It's given here. Okay. Then Robert May, global species diversity about 7 million. Alexander Humboldt, species area relationship. Here, students, they can ask you any of the following to tell you. Okay. Now, students, David Tillman did what, students? David Tillman found that the plots with more species showed less variation. For example, here I have one plot. Here I have one more plot. Students, which is your favorite plant? Students, which is your favorite plant, students? Which is your favorite plant? Quickly. Let's take my favorite plant is sunflower. So here I will grow only sunflower here. Only sunflower. Only sunflower. But in this I will grow sunflower, I will grow roses, I will grow lilies, I will grow everything here. Here we can find many plants. Here we can find many plants we can find here. Many plants we can find, students. Now tell me, students, in the chat right now. Tell me in the chat right now. In A or B, where do you find more variations? And it can keep on undergoing variation. It can undergo variation till die very soon. That is this one. Okay. So if there are many plants, it will show very less variation. See that? So plants with more species showed less year-to-year variations in total biomass. So less variation in terms of biomass. So what should we do, students? In every single time, in future, if any one of you become farmer, if you become a farmer, students, never only plant one type of plant. Always try to alternate. For example, always try to add a leguminous plant. Why? Can anyone tell me, students, why should we add leguminous plant? Because leguminous plant will do what? It will have rhizobium for nitrogen fixation. So leguminous plant, some other plant, leguminous plant, other plant. So always you have to mix and match. Mix and match is better. Okay. Now, students, Paul Ehrlich gave us the rivet-popper hypothesis. Again, students, can anyone tell me here what is actually rivet here? What is rivet equal to? Students, what is rivet equal to, students? What is rivet equal to? What is rivet equal to, students? Now, in 1992, Earth Summit, students, very important, Earth Summit held in Rio de Janeiro. All nations to take appropriate measure for conservation of biodiversity and sustainable utilization of benefits. Very important. 2002, World Summit. This is Earth Summit, this is World Summit on sustainable development. Okay, sustainable development, very important. Now then we have Fleming, Chain and Flore in 1945, Nobel Prize for Antibiotics Potency. That is from microbes chapter. That is from, students, microbes chapter. Done. That's all, students. That's all. So let me just call Gopika ma'am right now, students. Let me just call Gopika ma'am, and then ma'am will continue with ma'am's part. Students, how was it? How was the rapid fire? In 30 minutes, we finished the entire all the dates, all the scientists from botany in one shot. Okay, students. In one shot. Students, how was it? Students, quickly fire emoji in the chat, all of you. I want everyone to put fire emoji in the chat and invite ma'am. Quickly, students. I want ma'am to have full high Josh in the chat for all of you. So if you do not show the Josh, ma'am will not come. So I want everyone to put fire emoji in the chat to show the Josh, and ma'am will join eventually. Quick, students, quick.
Students, am I audible and visible to all of you? Hello, welcome to Vedantu NEET English. Yes, yes, please let me know if I'm audible and visible to all of you. Good evening, good evening. How are you all? Just give me a minute, okay? There was actually a small problem with this, Okay, yes. Good evening. Hello. Yes. So students, quickly share the link with all your friends. Ask them to join. Okay, ask them to join. I hope I'm audible and visible to all of you. Yes, okay. Perfect. Let's start the class. Okay, so I have very less scientist students. Okay, Basavaraj sir didn't give me any scientist. He's like, I want to do speech about scientists, so please give me all the scientists. Okay, so I am only left with molecular basis of inheritance and evolution. So yes, we are going to finish it in like 10-20 minutes. Okay, yes, I'm doing great. Thank you so much. Let's start. Okay, first we will start with molecular basis of inheritance. So we will start, I hope the brightness is okay. All of you are able to see this, I believe. So we start with Friedrich Mischer. He was the first person to talk about what, students? He was the first person to say nuclein, right? Nuclein. He was working on some cells, pus cells, and he noticed that there is something that holds all the genetic information, and that was the name nuclein. Okay, nuclein. Okay, and then students, one more person came after that, that was Altman, and he was like, wait, this nuclein has acidic properties, so why don't we call this nuclein as nucleic acid? Why don't we call this nuclein as nucleic acid? So this both people, please remember and keep. Okay, done. Okay, very good. Now James Watson and Francis Crick, he proposed the double helical model of DNA and he proposed the semi-conservative model of DNA. Who? Watson and Crick. They are like a combo, okay? They are like a combo, like how in our class combos come, no? How me and Basavaraj sir is a bio combo, like that. Okay, so it is proposed double helical structure and semi-conservative. Now, Wilkins and Franklin. Wilkins and Franklin were the people who told that with the help of X-ray diffraction, we understood that DNA is a double helical structure. Okay, we understood that DNA is a helical structure or a double helix structure. Okay, so that was Wilkins and Franklin, and with them even Watson and Crick also got recognized for saying that X-ray, with the help of X-ray diffraction, DNA is a double helical structure. Are you understanding? Will you tell yes? Huh? Will you tell yes? Will you put fire emojis? Will you put thumbs up? This has become like one small kindergarten class. Did you understand? Please clap your hands. So yes, if you understand, please put thumbs up in the chat box and put fire emojis in the chat box. Okay. Yes, double helical structure. Now, Irwin Chargaff, what did Chargaff say? That Chargaff told that when you talk about DNA, the ratio of A and T should be equal to G and C. Is it right? Not exactly, students. The ratio A to T. Okay. So look at the English. Okay. We are very poor in English. That's why we make mistake. It is A to T. And the ratio G to C. That means if I am trying to make a pair in a class, if there are 5 girls, I need 5 boys to make a pair. No, I cannot be like, okay, 5 girls are there, so 2 boys will play. No, right? So 5 girls and 5 boys is necessary. So he is telling that the number of purines should be equal to the number of pyrimidines. Okay, the number of purines should be equal to the number of pyrimidines. Yes, very good. That is the energy that I need. Now, Francis Crick proposed the central dogma of molecular biology and postulated an adapter molecule for translation. If you don't remember this also, it's okay. But please remember, Francis Crick was the person who proposed the central dogma. What is central dogma? All of you tell me, what is central dogma? Come on, quickly. Yes, AT is double bond, GC is triple bond. Okay, who is stronger? G and C is stronger. Okay, my name is there, G is there. So, okay. Yes, come on, tell me this answer, students. What is central dogma? Quickly answer. Yes, very good, very good. Ma'am, central dogma is where DNA forms mRNA, that's called transcription. mRNA forms, protein, that is called as translation. The whole thing we call it as central dogma. And DNA also can do self-replication, right? Yes, very good, very good. Is it lagging? Is this lagging or is it fine? Someone is telling lagging. I can't see any lag. Okay, now, Frederick Griffith, students, Griffith conducted the transforming principle experiment using Streptococcus pneumoniae and mice, and he used 2 strains. What are the strains? S strain and R strain, right? S strain and R strain. Clear? That's fine, right? Okay. There's no lag. Okay. You people are lagging. Okay. Brain is lagging. Yes or no? Brain is lagging. Huh. Okay. Now, Avery, MacLeod and McCarthy. So, even though Griffith told that let's call that as the transforming principle, Avery, MacLeod and McCarthy was like, no, wait. We need to find the biochemical nature of the transforming principle. That is, who exactly got transformed from the heat-killed S strain to the R strain. So, that is when who came, students? Avery, MacLeod and McCarthy. He conducted the experiment for biochemical characterization. Now, tell me, what were the things that they used? What were the 3 enzymes that they used? Come on, quickly in the chat box. Quickly in the chat box. Yes, flow of genetic materials from DNA. Brain is not there, it seems. Okay, good. Okay, I like your realization. Now tell me, biochemical nature was done based on which and all enzymes? Faster, answer faster. I think there's a problem with the live. Suddenly students are dropping. Wait, let me just check. Okay, this happens when we do it from 2 systems, students. Otherwise, it is fine. Okay. Okay, yes. Very good. DNAs, RNAs and protease. Okay. Now, Hershey and Chase students conducted the blender experiment, or we call it what? The bacteriophage experiment, Hershey and Chase experiment, where they used 2 things. What did they use? S35 and P32 to prove the experiment. That is, this is only present in protein, this is only present in DNA. So, if we see that the pellet has the radioactivity, that means it is confirmed that DNA is the genetic material. Okay. Now, Messelson and Stahl students experiment proved the semi-conservative model. That means what? Parent. Okay. Parents try to solve kids fight. Okay. These are the daughter. So how will they form this? By using it as a template, okay? So here what is going to happen? You're going to get a hybrid. That means one is going to be a parent strand, one is going to be a daughter strand. So you're going to get a hybrid. One N14, N15. Messelson and Stahl proved it with the experiment. Clear? Yes, excellent, excellent, excellent. Next one. George Gamow suggested that coding for 20 amino acids, the code should be made up of 3. That is, he told that we'll try 4:1. It did not work out. Let's try 4:2. Did not work out. Let's try 4:3. And yes, it finally worked out, because out of the 64 codons, 61 codes for amino acid, 3 are stop codons. Okay? Excellent, students. Now, Har Govind Khurana developed a chemical method in synthesizing RNA molecule without the defined combination of bases. That means he did homopolymers U. He did copolymers UA. Okay, it will look like I'm some that tribal singer, no, UA. Okay, it will sound like from third person comes into the student, into the studio, they'll think, ma'am is singing some tribal song or what. Okay, yes, they did cesium chloride, Messelson and Stahl. Now, Har Marshall Nirenberg, okay, he developed a cell-free system. Okay, he took a cell-free system for protein synthesis. So, one person without the actual triplet codon, they found out how we can synthesize mRNA, okay, synthesize RNA. Another person tried to do how we can synthesize protein without an actual cell. Okay, without an actual cell. Clear? Yes or no? Now, Jacob and Monod first elucidated lac-operon model. Okay, Jacob and Monod first elucidated lac-operon model. That is, in the presence of lactose, the gene is going to be switch on. In the absence of lactose, the gene is going to be switch off. Clear? Yes or no? Very good. Okay. Now, Frederick Sanger, students, he came up with the Sanger sequence. That is, sequencing the DNA fragments in an automated DNA sequencer. Developed a method for sequencing amino acid in protein. Okay. Sequencing amino acid in proteins. Clear? Sequencer. So his name, that name is called Sanger sequencing. Okay, Sanger sequencing. Clear? What and all I have to do for you people, I have to explain all this, no? So I have to sing tribal songs, otherwise survival won't happen. So I have also put the dates here. Okay, look at the years. 1869 Frederick Misher. Okay, 1869 Frederick Misher. James Watson and Crick 1953, Irwin Chargaff 1950, Frederick Griffith 1928, Avery McLeod and McCarthy, almost a bigger span, okay, and Hershey and Chase 1952. Okay, so the same thing I have put in boxes here so that you can read it from here as well as from there, because dates are mentioned here. Okay, dates are mentioned here. Now, next is evolution chapter, students. Now, evolution chapter, the main people are Darwin and Louis Pasteur. So, Louis Pasteur disapproved theory of spontaneous generation. Okay, disapproved theory of spontaneous generation. You don't have to know the names of every single person, Reddy, Splansky, no. You just have to know Lamarckism, Darwinism, that's it. Okay, so here they won't ask you questions on every single thing. Clear to all of you? Now, look here. Louis Pasteur disapproved theory of spontaneous generation, that is abiogenesis, and he demonstrated how life comes from pre-existing cells or pre-existing life. Clear to all of you? Excellent. Now, Oparin and Haldane, very important. Okay, Oparin from Russia, Haldane from England, proposed that theory of chemical evolution. Theory of chemical evolution. S.L. Miller, he did an experiment to prove the theory of chemical evolution, where he took a flask, he put what and all, students, here? Question comes from here. CH4, NH3, H2O, H2, and he gave 800 degrees Celsius with the help of electrodes to actually prove the experiment. Correct? Okay. Yes, Carl Linnaeus gave two kingdom classification. What happened? Okay, what happened? Yes. Now, students, Ernest Haeckel proposed the embryological evidence of evolution. Very important. Ernest Haeckel also came up with the three kingdom classification, right? Now, Carl Ernst von Boyer, so he rejected. One person proposes, another person rejects it. Okay. So, here Ernest, here also Ernest. So, even though they both were really honest, still they both proposed and got rejected. And think about you people, students. Okay. So, Ernest Haeckel proposed embryological evidence of evolution. Ernest Haeckel von Baer rejected rejected embryological evidence of evolution. Okay, clear to all of you? Yes or no? Is your hand aching because of putting the fire emojis continuously? Huh? This is a chill class, only 1 hour class. Why are you taking tension? Huh? Why are you people taking tension? Be relaxed. This and all easily in 15 minutes my students can study, because this is all theory. Okay, this is all what you study in theory. Molecular basis also what you study in theory. Only dates you have to go through. Correct? Yes or no? Arey ma'am, some of them were tube light, no? So after some time it's clicking for them. Wait, ma'am, did ma'am just roast us? Okay? No problem, mock test is important. Yes, okay. Yeah, you can tell I am a student who is preparing for NEET and reject them, okay? Because they have to wait, no? For 5-6 years, so they will just go by themselves. Okay, now, yeah, okay, now this has become destroying, okay? I've started destroying, not roasting, destroying, okay? So let's go. Now, come on. Lamarck students propose the theory of inheritance of acquired characters, okay? That is called as Lamarckism, right? Now, I think I can make a season of roast, right? Like, you know, I can make one whole episode of where and all I have roasted you people, right? But I don't know where and all I have roasted. It comes naturally. So, I feel at this point, after your NEET, no, if you're feeling very bored, what to do in life, okay? I know counseling will take all your life, but still, before that, if you feel like, okay, I have so much time in life, we should definitely make a roast video, okay? We should make an entire episode of Gopika ma'am roasting students. Come on. Okay, Lamarck proposed theory of inheritance of acquired characters, so that is called as Lamarckism. Now, students, Darwin, he did multiple things, okay? He's like that person who does, you know, who is on this channel, then on that channel, and this channel, and this channel. He's everywhere. So we cannot put him in any of the, you know, those, you seen one fellow who has got 99% will be everywhere. Okay, he'll be in all the places in the world. He would have studied from all the institutes. I really want to ask him, how does he do that? Same way Darwin is. He went to Galapagos Island and he found Darwin's finches. He took his HMS Beagle and he went, so that also we have to study. Then Darwin told that natural selection exists, so we have to study about that. Then Darwin was having a competition with Hugo de Veris, telling that, you know what, I am doing this, working on evening primrose, and he spoke about variation. Okay? Yes? Clear? Okay, very nice. So students, you have to remember about Darwin, and also you have to remember about Hugo de Veris. Okay, whom will you remember? Hugo de Veris. Okay, so we will come to Hugo de Veris. Okay, yes, let's go forward. Don't you people also think that way? How is the student studying from every place? Huh? I think that way. Okay, I always think, how? Okay. Now, Lamarck proposed the theory, understood. Darwin students proposed the theory of natural selection, which is called as Darwinism, which is one of his most important one, like the actual institute he studied from. Okay. Alfred Wallace proposed natural selection similar to Darwin's conclusion, because both of them read Thomas Malthus' book. Okay, book, or like it could be like a documentary or just a information. Okay, Malthus. Clear? Yes, I will definitely share this PDF. When have I kept the PDF for myself? I will definitely share the PDF, okay? Yes, Thomas Malthus, this person, okay, studied population, and his idea influenced Darwin as well as Alfred Wallace. Now, Hugo de Very students proposed mutation theory of evolution. All of you tell me, on what did he work on? On what did he work on? Come on. Thank you, Shailaja. What did he work on, students? I wonder same as you wonder. Wow, Levi. So we both are this wonder woman and wonder person, huh? Yes, that's my plan. Future plans is that, to start a museum and draw my own animals. Now tell this answer now, otherwise I'll scold you people. Hugo de Veris did mutation theory of evolution. He worked on what? Yes, very good. He worked on evening primrose. Okay. Thomas Malthus, you don't have to remember, but remember that Darwin as well as Alfred Wallace. Now imagine, students, my notes. Okay, I wrote about population. Okay, let's take example Krishna and Anupam. Krishna and Anupam read about it, and they worked about it, and they made two different theories. So I am Thomas Malthus. Okay, Thomas Malthus, Anupam, and who did I like? Krishna. One is Alfred Wallace, one is Darwin. They got inspired by my work. Understood? That is what. Okay, evening primrose. Very good. Okay, evening primrose. They will ask you this question. Okay, next one. Hardy-Weinberg proposed, obviously students, Hardy-Weinberg principle will be proposed by Hardy-Weinberg only, no? So he proposed the Hardy-Weinberg principle about genetic, okay, Hardy and Weinberg, okay, two people. Hardy and Weinberg came up with Hardy-Weinberg principle about genetic equilibrium, that if everything goes well in my life, then I am going to become successful, like that. Okay, yes, very good. Excellent. Okay, excellent. So since sir took all the scientists from me, I'm left with very few scientists. So let's just respect them and move forward. Stanley Cohen and Herbert Boyer, students, they proposed the first recombinant DNA technology, that is your rDNA technology, where we insert the foreign DNA into a plasmid, okay, plasmid or a vector, and insert it into the host organism. Clear? Yes. Very good. Very good. Next is some dates. Okay. Again, all the dates are from molecular basis of inheritance. 1928, Frederick Griffith. 1944, Avery McLeod and McCarthy. 1952, Hershey and Chase. 1953, Watson and Crick. 1961, Nirenberg. Okay. You don't have Matai, you only have Nirenberg. The first codon of genetic code. 1977, students, Sanger sequencing. 1990, initiation of human genome project. 2003, these two dates are very, very important. Okay, among all the dates, these two dates can come as a question. These dates, they will ask you the experiment more than the date. Okay, so initiation of HGP 2003, completion of human genome project, which is very, very important because this proves that HGP was a very big, okay, it was a mega project. It took so many years to complete it and so many people were involved in it. Okay, done, done. Yes or no? Yes or no? So I've just typed that here also. So you can see 1990 HGP project started. 2003, that means almost 13 years it took to complete the project. May 2006, students, the sequence of chromosome 1 was completed only on May 2006. Were you guys born? Were you guys born? 2006 you were born, huh? Now don't tell me, ma'am, please calculate the maths and you tell if we are 18 years. Not possible. That much maths if I knew, I would be in some IIT. Now tell me, tell me. The sequence of chromosome 1 was completed only on May 2006. You were born. Okay, don't be like, oh ma'am, please ma'am, other people will know my age. You're not any ancestral property, so you can tell your age. 2008, see, all young people than this there. Okay, very good. Some at least are born. Okay, the sequence of chromosome 1 was completed only in May 2006, 1 year before your, before your birth or after your birth, it was found. Clear? Fine? 2006, okay, fine. Now in 1930, don't reveal your age. They will know how many drop years you took. Don't reveal your age. Leave it. Leave it. Then don't tell later, ma'am, this fellow texted me asking, how is your experience with this many drops? Let's not go there, okay, students? Let's not go there. Okay, 1938, evolution, coelacanth fish. This again, please try to remember. 1951, family planning program, that is RCH, okay? Family planning program was done. Again, reproductive child health care. They were telling that, okay, you have to do family planning. We need to control the population, okay? 1971, reproductive health legalized MTP. Okay, reproductive health, that is legalizing of MTP. Clear? Yes? Okay, very good. Very good. Now, AIDS first reported was in 1981 and again it was in a gay community and people thought it was to something to do with the sexuality, but it was not. Okay, it was not. Okay, clear? Now, 1983, students, Eli Lilly company, okay, Eli Lilly company, an American company, prepared 2 DNA sequences corresponding to A and B. That means they found the humulin. Okay, they found the humulin. That is very important. Eli Lilly company found the humulin. Okay, humulin. Yes. That also can happen. This not, they might not be taking drops. Okay, they might have sat in LKG, UKG two times. They have double LKG and double UKG. Happens because age does not matter. No, now in the sense now age matters a lot. Like you have to be 6 years, you have to be 5.5 years, something like that is there. So that time they'll have double UKG and double LKG, which you cannot write it in your resume. Okay, let me tell you, it cannot write it in your resume. Got it? Yes, very good. Eli Lilly company, will you forget? No, ma'am, we will not forget. Okay. Now, 1997 students, Rosy cow. Okay, transgenic animals, that is lacta-albumin was found. With this only, please remember alpha-1 antitrypsin, that is emphysema. Okay, emphysema. Yes. Now, 2002 students, biopiracy. 1950, the whole plant could be regenerated from X-plant. From where did they put this? They put it from strategies. Okay, strategies chapter they took in. They added it here after deleting the chapter. They were like, oh, let's just delete the chapter, but let's put all the major chunk portion into biotechnology. Okay, what a smart move. That whole plant could be regenerated from an X-plant. It was found in 1950. Just go through these dates. Okay, I'm not telling dates question will 100% be asked, but just go through all these dates. Okay, clear to all of you? 1990 students, the first, this is very important. Very important. 1990 kid, severe combined immunodeficiency. How did they find it out? In a kid who was 4 years old. Okay, in a kid who was 4 years old. So the first clinical gene therapy was given in 1990 on to a 4 years old kid with adenosine deaminase. Okay, 1997 student, an American company got patent rights on basmati rice through the US Patent and Trademark Office, which was actually taken by a dwarf, semi-dwarf variety from India. Correct? Yes or no? Yeah. Full dark. Okay. Neat, dark, emotional damage is happening. Students, all this is jokes. Okay. Take jokes as jokes. Don't take it like a case. Don't make it a case. Right? Yeah. Okay. Yes. Clear? If you don't, I always tell, no, in my class, we have to be light-headed, not light-headed, light-hearted. Okay. Head is light means then it will become problem for me to make you understand. But otherwise, let's take everything. This is a very simple class. Okay. So yes, we had so much fun. Now students, listen here. In all the VLC centers, you are going to get 40% scholarship, okay, on all the NEET and JEE courses. And I would say it is a very good thing for 2026 aspirants, okay. And we are also going to come and meet you on May 11th and mostly on May 17th, if I'm not wrong. If not 18th, on 17th, Saturday. Okay, so May 11th in Chennai, Anna Nagar Center, and 17th in Hyderabad. So definitely meet us. And also if you're planning to take, okay, a course, that is, ma'am, I want to take a course, then please go and avail the 40% off now. The link is in the description box. Okay. Yes. Link is in the description box. Okay. Yes. Manasvi, I see your full, like she puts comment like, it's like full fire, full hearts and full glow, everything is there. Okay. Yes, come on. ISER classes we have to see, students. Yes. Okay. Yeah, very disheartened with the incident, dear. We can only pray for them. We can't do anything. Okay. Yes, exactly. Okay, thank you so much, students. That's all. So please make sure that you do this availing. Okay, if you're a student who is watching this video for 2026, do not waste 40%. You understand, no? 40% in a VLC center is very, very big of a number. So please make sure that you reduce that much burden on your parents and go to the nearest VLC center and avail this offer. Okay, and all these notes will be available on Telegram channel, students. All these notes will be available on these 2 Telegram channels. Please make a note of Telegram channel so you will be able to get the notes. Okay, Vedantu NEET English. Okay, name of the channel only, Vedantu NEET English. And if you want zoology notes, then it is Gopika Biology Official. Okay, who is the official? I am the official. Okay, official. So you will get all the notes in these 2 Telegram channels. Okay, so join the Telegram channels and see you all on May 11th and May 17th or 18th. I will confirm the dates with you. Okay, so if you are in Hyderabad or Chennai, do not miss the event at any cost. I am so excited, students. If you can travel, like if you have any of your aunt, uncle who stays close by, you can come there and stay for one day and come and meet us. That would be great because I have only heard your names and I have already connected with all of you just by your names. And if I want to meet you all real, I think it's going to be so much happiness. Okay, so do not miss it. Okay. Thank you so much. Thank you so much. Vizag. Yeah, I know I'm getting a lot of requests on Vizag. I really want to come to Vizag, but I do not know how much distance it is there. and all of that. So we will check and let us know. Don't worry, we will come some other time as well. Okay? Yes. Yes, quickly smash the like button as well if you haven't yet, and subscribe to the channel, because this is the best English channel possible, where we have completed the syllabus prior and we are also doing question practice for all of you. So I want all of you to definitely subscribe to the channel and smash the like button, and please let us know in the comments how the class was. I really enjoyed the class. I hope all of you also enjoyed, because after a long time we are having a 1 hour class. Okay, so this notes will be available on this Telegram channel, and for those students who do not have Telegram channel, the link will be updated in the description box. Okay, link will be uploaded in the description. Clear? Yes, students, thank you so much. Bye-bye. Chikbalapur, I'll come like that only, Lokesh. It's closer to me. I'll come like a travel. Okay? Yes. Northeast. Haha. You people crack NEET and you take me for trip. Okay? Company will tell, excuse me, ma'am, sit at home. Okay? You people take me for trip. Yes, okay. Bye-bye. Definitely drop down in the comments how you felt the session was. Okay? Bye-bye.